Good morning, good evening. Welcome, everybody. We're so pleased that you're here for this inaugural webinar for our new section of the Frontiers in Systems Biology Journal. I'm Dr. Jennifer Lovejoy, and I'm going to be one of your hosts for today. So we'll just give this another minute or so to let people continue to join, but we'll get started here in just a moment. All right, well, I think we will go ahead and kick things off. And again, welcome to everyone. I know we have people joining from all over the world, so thank you for taking time out of your day, evening, night, wherever you may be. And we are so excited about this webinar that we are offering this morning with Dr. Lee Hood. We'll be getting into that shortly. But before we do, we want to say a few words about our new section and about the journal. And so to begin that, I am going to turn things over to Professor Yoram Vodovotz, who is our chief editor of the Frontiers in Systems Biology Journal. Yoram.
Thanks, Jennifer, and thank you all for taking the time to attend this, what I'm sure will be a fantastic webinar. As I'm sure you're well aware, research in biology and biomedicine is tackling ever more complex questions than ever before. And our goal here at Frontiers in Systems Biology is to help address this complexity in an integrated and interdisciplinary fashion, really aiming to cut across entrenched silos, which exist even in our own supposedly synthetic discipline. And so our journal consists of 6 sections currently, and they are data and model integration, evolutionary systems biology, integrative genetics and genomics, integrative systems microbiology, integrative systems neuroscience, multi-scale mechanistic modeling, systems and synthetic immunology, translational systems biology and in silico trials, and the section that is headed by Jennifer and Sean Manion, who will introduce more about this, is systems concepts, theory and policy in biology and medicine. And this section is really intended to act as a bridge across all of our other sections, and hopefully to stimulate perhaps a greater degree of conversation and thought about how to actually do the science of systems biology. And so as such, it's my privilege and pleasure as the field chief editor of Frontiers in Systems Biology to host really a pioneer and central figure of our discipline, someone who certainly influenced me tremendously in my career, Dr. Leroy Hood. And now I'd like to turn this over to Jennifer and Sean, who are going to introduce Dr. Hood and also the section. Thank you.
Thank you, Yoram. And Sean, I'll kick it over to you.
Excellent. Hello, everyone. My name is Sean Manion, along with Dr. Jennifer Lovejoy. We are heading up this new section of systems concepts, theory, and policy in biology and medicine. This is part of, as Yoram noted, the bridge across many different areas of Frontiers systems biology and systems biology and systems medicine in general. As you all know, science itself is oftentimes specialized and becomes a bit reductionist, and with that, systems thinking brought to biology and medicine has been very helpful. But sometimes the systems thinking needs to be more broad than that, and considering policy, considering concepts to bridge all of these different areas together is the goal of this journal. My background is one of a bit bridging together a lot of things. I started in economics and moved to biochemistry and then neuroscience. I got my doctorate looking at military medicine at Uniformed Services University, but quickly wanted to look at a bigger picture of how science got done within the federal government and reaching out to academia and industry. So I spent a long time in the federal government overseeing networked research for traumatic brain injury, looking at how to integrate not only the science and the research, but how to bridge that into the knowledge translation pipeline, and how that came into play with regard to policy and how policy impacted the advancing of the science. I moved into the industry side of things, where I am now the chief scientific officer of a company called Quidium Health, focused on bringing emerging technology to health and life sciences. And it's there that I've become very interested in how to bring systems thinking to not only the science, but to the technology that we're bringing to help us with the science. And that's what I hope to do with this journal. With that, I'll hand it off to Jennifer to give you a little of her background and focus on the journal as well.
Great, thank you. Yeah, it's such an exciting opportunity, really, to have a journal that is going to focus on integrating all of these pieces. So from my perspective, my background, I have a PhD in physiological psychology, and so in some ways, that already was a systems approach, bringing together the psychological and biological sciences. Of course, a lot of neuroscience, similar background to Sean. But my early research in academia was really focused on conditions, diseases like obesity and type 2 diabetes, where there is a clear integration of both physiological, genetic, biological systems and behavior, psychology, emotions, and other factors that have to be considered together before you can really start to effectively deal with some of the chronic conditions, which of course are the things that plague our health system the most today. So as I went further in my career, I left academia and went into industry around 2008 to look at how do we apply the evidence that we are getting from these clinical trials and actually bring them into the real world and help people. And I had the privilege of meeting Dr. Hood in 2013 when he was starting to think about a vision, which we'll talk about more later, of P4 medicine and scientific wellness and how you bring that into our current system and what are the challenges and opportunities there. So I joined the Institute for Systems Biology in 2014 to help run a clinical trial called the Pioneer 100. And then subsequently, we spun out a company called Aravale, where I was the chief translational science officer, bringing together behavioral science and multi-omic data to help people with optimizing wellness and preventing disease. So it has been a really amazing journey, and I am thrilled to be here with the team. I am excited about the launch of this journal, which I hope will really not only bring in the engineering, the physiological, the genetic side of things, but the behavioral and the environmental and the regulatory, as Sean said, which is such a factor in how we bring this into medicine in particular, to actually be able to use the advances in systems biology and systems medicine in clinical practice. So those are some of my interests. And with that, I am going to introduce Dr. Hood, who, as I said, I've had the pleasure of working with for almost 10 years now. And boy, I tell you, that I had a couple minutes here on the agenda to introduce him, and Lee is actually one of those people that it takes several minutes to introduce because his career has been so stellar. Obviously, a world-renowned scientist, often considered to be the father of systems biology, but has had quite an amazing career. I'll start sort of close to the beginning. We'll get into this a little bit more, but he got his MD from Johns Hopkins University School of Medicine and his PhD in biochemistry from Caltech. He went on from there to join the National Institutes of Health, where he worked in the immunology branch of the National Cancer Institute as a senior investigator. And then he returned to Caltech as a professor and eventually served as the chair of the Division of Biology for about a 10-year period. And it was during this time that Lee and his colleagues developed a number of key instruments that have really changed the face of science and biology, including sequencer synthesizer instruments that ultimately paved the way for the Human Genome Project that mapped the human genome much later than those instruments were developed, but could not have been done without that. Let's see. And then he left Caltech to go to the Department of Molecular Biotechnology at the University of Washington, which was unique in that it was the first true integrative department, trying to bring together biology and engineering and chemistry and these other disciplines into one department, cross-disciplinary biology. And still, that was not quite the systems biology that Lee was hoping for. And so he co-founded the Institute for Systems Biology here in Seattle in 2000 and served as its first president from 2000 to 2017, and is currently a professor and chief strategy officer there. More broadly, Lee was a recipient of the National Medal of Science in the United States, which is really one of the highest honors that can be given to a scientist in the US. He's a member of all three of our national academies here, the National Academy of Sciences, the National Academy of Engineering, and the National Academy of Medicine. And of the more than 6,000 scientists who belong to one or more of these academies, Dr. Hood is only one of 20 people who belong to all three. He has also played a big role in industry, founding 17 biotech companies, including Amgen, Applied Biosystems, Aerovale, and NanoString. He's published over 1,000 peer-reviewed articles. He holds 36 patents. Again, we could go on and on, but Lee, what an honor to be with you here today. And we're going to talk about some of the milestones in your career and really what you have seen about systems biology as it has evolved over the course of your career. But I want to start back in the beginning. And as I think about science, one of the most important things for all of us actually is mentorship. And we get different levels of mentorship, I think, at different points during our lives and during our careers. So I want to go back very early. You grew up in Montana, in rural Montana, where probably not a whole lot of biotech going on in those days. But were there any early influences in your life, mentors or other factors that you see now in hindsight kind of moved you in the direction of the systems thinking that you have pioneered?
Well, I would say there were two opportunities that really moved me towards systems biology. The first was the fact my father was an electrical engineer for AT&T and responsible for a statewide microwave system. But what he did every summer was teach AT&T employees about engineering. And as his son, he wanted me to take these courses, which I was a little reluctant to do at first. But in retrospect, courses on systems engineering and integration of component electrical parts, all of these engineering ideas set a framework for my thinking, which was different from traditional biology. And I thank him for that. So that was the first. But the second was the fact that my granddad actually built a camp in the Bittertooth Mountains in southwestern Montana for geologists from the east. So geologists and their students in the summer from Columbia and Princeton and Harvard and Yale came out, and I ended up taking courses with these students there. And I remember the summer before my senior year in high school, I actually mapped an anticline. And what those scientists taught me was the importance of understanding in enormous detail aspects of geology in this case, but the mapping of an oil anticline showed the systems aspects of it, where you had to put together all the different formations in order to explain just why oil was found in that particular location. So those two set me to, I would say at that time, inchoately thinking about systems biology, but they both very much influenced the biology I was to do later.
Wonderful. So I've heard you speak, and often in your talks, you'll mention 7 paradigm shifts that you have seen and experienced in your career. And we may not have time today to touch on all of them, but I think a number of them are really quite important for our audience that is here today. And so I did want to touch on a few of them. And I'm going to go back, start with the Caltech days when you were working on a number of different instruments, and you had the idea of integrating engineering principles with biology. And so maybe some of what you just said about your early experiences led to that, but what were some of the insights that you had along the way about the importance of integrating, and how are you applying that work today in some of the AI approaches that you are looking at?
Well, I think what really got me started at Caltech in technology was sitting down at the beginning, as assistant professors do, and say, where do I want my career trajectory to go in the future? I really had an interest in human biology and actually human disease, and I was overwhelmed by its enormous complexity. And that's when I started thinking about, how can we deal with this enormous complexity? And I came up with 3 ideas. One idea was big data. That is, we had to create the means in an individual human being for assembling many different kinds of data. A second idea was the idea that blood is really a window into health and disease because it bathes all of your organs. They secrete molecules into it. And if you quantify those molecules, they can serve as reporters for the health state of those organs. And that has turned out to be true. And of course, the third idea was somehow, when we aggregated all of this data from these diverse sets, we had to be able to think about integrating it together to create a whole that assembled diverse types of information into models, in this case, about human wellness and about human disease. And of course, a fourth avenue that came later that was really important was the avenue of machine learning, of contemporary AI. And indeed, today we use knowledge graphs, we use digital twins, we are using large language models, all of the sophisticated ideas of AI to be able to look at the complexity of a human being and begin assessing for each individual how we can optimize wellness and avoid disease. So this is the big new thrust that we're pointing toward for the future.
Yeah, absolutely. So let's talk about the Human Genome Project. You are well known for your involvement with that, and I know that it was not an easy thing to convince the government to get involved in. So what were some of the experiences that you had along the way there to both the challenges as well as the ultimate obvious success of the project that might inform other people who are really thinking about doing big integrative science as you have done?
Well, I think this all got started in the spring of 1985 when Bob Sensheimer decided to invite 12 scientists to Santa Cruz to assess the Human Genome Project. And our analysis was absolutely fascinating. First, we concluded it was doable, although very complicated at that point in time. And second, we were split 6-6 on whether it was a good idea. And those opposed were primarily opposed because this was biology's first big science problem. And they were terrified that the funding for big science would take away from small science, which at that time was the traditional kind of biology that was done. And of course, in retrospect, this turned out not to be true at all. Rather, the genome opened up incredible new areas of biology that all the R01 type science, the small science, has taken enormous advantage of. And I would say the Human Genome Project really did 2 important things. One, it developed automated DNA sequencing to a point where we could do any organism's genomes, and that has transformed so many different areas of biology, especially microbiology and so forth, in that we can now start from the genome as the basic source code for taking a systems approach to understanding the complexity of any organism you want to study. And of course, the second thing it did is for the first time it gave us access to human genetic variability and the ability to correlate that variability with wellness phenotypes and disease phenotypes. And of course, this is just now beginning to be a major feature in transforming our approaches to human health, as you well know, Jennifer.
Yeah, absolutely. So what led you to start the Institute for Systems Biology? What were you finding in your experiences at Caltech, at University of Washington, and within the industry examples that you kind of had your fingers in a lot of pies, but you chose to start this nonprofit research institute. How did that happen?
Well, when I was at Caltech, my lab did two really major things. One, I continued molecular immunology, which I started studying at my PhD at Caltech much earlier. And two, as a consequence of wanting to get into human biology, I developed a very large lab that had technology as a central thrust, and we developed machines for automating the sequencing of proteins and genes and automating the synthesis of those. And in fact, I'll say we went on later to add two different instruments, the inkjet technology instrument that synthesizes very large scale fragments of DNA, and the nanostring approach, which I think most people are familiar with. And this technology, as I said, made my lab big. And that was absolutely an anathema to the senior scientists in biology at Caltech, because biology was the essence of small labs, and they hated the idea of a big lab. And they argued I should be moved to engineering, and the chairman, fortunately, didn't make that move. But the second thing that happened is I got very involved and committed to the Human Genome Project. And again, biology was very unenthusiastic about the Genome Project, as many excellent departments were at that point in time. And because two of the things I was really interested in, the genome and technology, weren't very popular there, that's why I decided to take advantage of a Bill Gates offer and move to the University of Washington to start this first cross-disciplinary department there. And I went with the idea of wanting to build on top of this cross-disciplinary department, which was enormously successful over its 8 years of existence. And it really pioneered the fundamental software of the Genome Project. It pioneered the whole new field of proteomics with John Yates and Rudy Abersol, two of the real pioneers in proteomics. And we developed the inkjet technology for DNA synthesis there. But I was really unhappy because systems biology really required many things that biologists hadn't done in the past. For example, I remember going to the head of computing at the University of Washington saying, look, I really needed space and some assistance in building a strong computing entity within the department. And he came over and showed me a very small janitor's room and said, that's all the space any biologist will ever need to do computing. And, he just didn't understand that the world was going to change in really major ways. A second example was the dean at that time and the president of the University of Washington at that time argued that I'd never be able to approach Bill Gates without their permission because they had designs on what they wanted to get from him. And I argued, look, I knew him better than virtually anybody else at the university. I would know the kind of things that, and it made no difference. And I realized that if you're really going to start something serious, it wasn't, and it came to be a fundamental policy of mine. Really original new ideas can never be born in the midst of old bureaucracies. Almost always, you have to start something new to give them a chance to be born. And of course, systems biology was something new, and resigning in 2000 and creating the Institute of Systems Biology opened up this really exciting possibility of thinking in an entirely new way about biology. But I will say, I had a really close colleague at MIT that just before I started ISB said to me, you know, Lee, the systems biology is all hype. It's ridiculous. Don't waste your career on this kind of thing. And of course, that goes to show you how conservative scientists are in thinking about new options and new opportunities.
Yep. Well, we're going to come back in a moment to your policy about the difficulty of starting new things in existing systems. But before we go there, when I think about the founding of ISB, and one of the things that I know is true about you and your career, you are absolutely the most optimistic and persistent person that I have ever met. And I think that it is important to have both of those characteristics in science, and particularly if you're trying to do something important and something big. So when you founded the Institute for Systems Biology, of course, you had to find ways to fund it. And because you were looking to big science, you needed some big ways, some non-traditional ways to fund it. And ultimately, you ended up getting the government of Luxembourg involved. And so can you tell a little bit about that story, and how you sort of approached that with ultimately great success? But I think because of that persistence and optimism that kind of brought you to like, wow, what are we going to do here? So I think it's a fun story for people to hear.
Well, I think one of the most fundamental characteristics for people who want to start new things is determined optimism. You have to be positive. You have to be and determinedly positive in the face of a lot of criticism. Well, the story of Luxembourg is a fascinating one that started about, Luxembourg went from roughly 2006 to 2011. It was a 5-year program for $100,000, $100 million, $20 million a year. And it started when I really am a missionary of biology, and I decided it would really be cool if I could persuade a country to take on systems biology at that early stage. And I just happened to have a letter from a Jewish American fundraiser who said, look, would you be interested in starting a systems biology institute in Israel? So I went and spent a year going to Israel. I had visited its 7 academic institutions. I found one of them, Beersheba, that was very interested in setting up the institute. They actually drew the designs of the building that we would have there and everything. And at the 9th hour, just as we were getting ready to start it, this Jewish American fundraiser called up and said, well, I can raise money for the Jewish Institute, but I can't raise money for ISB. And the agreement had been that he would raise $100 million for each of us. So that was an enormous disappointment and made it impossible to go on. But without going into detail, I next went to Ireland at the invitation of a former NSF friend of mine that was manager of the science and technology program. He wanted to do something similar in Ireland, so he invited me to come and talk about systems biology to the 4 major educational institutions there. I did so. And 3 of the 4 were really enthusiastic. And the 4th, that arguably the best of them, turned out to be skeptical about needing ISB at all. And in the end, they said they would enter a competition to make this institute, but they didn't want to do it with ISB. So I closed that thing down. And I'll just say I did some with the Heritage Foundation in Alberta. And all I can say is, in dealing with the Canadians, it was death by democracy. Nobody could ever make a precise decision. And my final attempt was with South Korea. And there we really had a beautiful government program set up to have a major systems biology institute. And at the last minute, they changed parties, and the new party came in and canceled all the major projects of the old party. So when I went to the chair of my board, Roger Perlmutter, a very famous pharmacology pioneer and leader, he said, for Luxembourg, I got a letter from a friend of mine at an institution who said Luxembourg is looking to start something quite serious in biotechnology. Would you be interested in help? Roger said, nope, I forbid you to go. You've wasted your time 4 other times. And I said, nope, I'm going to go, Roger. I'm going to try at least one more time. And Roger said, well, if you succeed, I'll believe in the tooth fairy. So anyway, we went to Luxembourg. What was different about Luxembourg from all of the rest is from day one, the major person we interacted with was the Minister of Economics, who was really driving this drive toward biotechnology. And after talking with him on the phone initially for an hour, he said, okay, write us a proposal and tell us how you'll do it, and we'll move ahead. I said, what are the boundary conditions? And he said, none. Ask for what you need. So I asked for 100 million over 5 years. That's exactly what they gave us. And that money was utterly transformational for ISB in terms of recruiting new people and in terms of doing research on projects that I really had trouble getting funded at NIH, which has become in many ways quite conservative. But this absolutely transformed ISB. And the chair of my board wrote me an email that said, I believe in the tooth fairy, when I told him that we'd succeeded in doing this. So determined optimism is absolutely essential.
Yep. Well, you know, and it strikes me the beautiful thing about that story is not only the ultimate success with Luxembourg and what that did for ISB, but as you were going around the world talking to all of these other countries and all of these other scientists, you were planting the seeds that may not have paid off for ISB, but now there are systems biology institutes in many of those countries, and there are systems scientists in many of those countries. So, so it really did help to seed, I think, part of the global movement in systems biology. So it's a fantastic story.
And I'll just say that the Luxembourg Institute for Systems Biomedicine is one of the leaders in systems biology in the EU. So we did succeed to that level.
Yep, yep. So in the last part of our discussion here, I want to turn to systems medicine a bit more. So you have described the future of medicine as P4 medicine and how that is essential. So I'll let you just briefly talk about what is P4 medicine and why do you think that it is the medicine of the future?
Well, in the very early 2000s, we started focusing our attention on healthcare and what we needed to do vis-a-vis systems thinking. And one of the first concepts we came up with was the concept that medicine should be predictive, preventive, and personalized. And then a year or two later, we came up with the idea that it also should be participatory. And we've come to the conclusion that the most effective way for achieving P4 healthcare is to take this data-driven approach to optimizing the health trajectory of each individual that was reflected in the Airvale 5,000 individuals whose data we collected as longitudinal clouds that gave us deep insights in how to optimize wellness, how to avoid disease, and so forth. But I will say the first 3 Ps, the prediction, prevention, and personalization, are science. And I think we really know now very well how to deal with those 3 Ps and very powerful strategies for them. The 4th P, which is participatory, I think is the big challenge we face today. And by participatory, it's exactly what we were just discussing. how do we persuade the stakeholders in healthcare to think about a data-driven health? How do we persuade the patients? How do we persuade the physicians? How do we persuade healthcare leaders? How do we persuade regulators? Ultimately, how do we persuade politicians that these are going to be important and transformational new initiatives? But I think P4 medicine captures the four essences of what this new data-driven healthcare is all about. And one of the initiatives I'm pushing through a nonprofit started several years ago called Phenome Health is the idea that we need a second major genome project, a project where we, through government support, analyze 1 million people over a 10-year period with regard to their genomes and their phenomes. And what this will do is two things that I think is going to lead to a big revolution in medicine. One, it will unequivocally demonstrate an enormous increase in quality of health for each individual. And two, it'll show exactly where we can go to save literally trillions of healthcare cost dollars by preventing chronic diseases in ways that we've come to understand. And this kind of revolution, I'll argue, is going to be the biggest revolution ever in medicine, a revolution in which we replace our disease-oriented medicine and our focus on the only way you make money in medicine is through disease, with a wellness-oriented and prevention-oriented medicine where we make money in the appropriate way by increasing the health rather than just by dealing with the disease.
So on building on that, I want to go back to one of your earlier statements about how you have made it sort of a personal policy that nothing new, nothing big can be created in an existing system. Obviously, the healthcare system in the United States and in most other countries is a pretty well-established bureaucratic system that is very difficult to change. And so, when we launched Aravail in 2015, the idea there was we would go direct to consumer. We would kind of take it out of the healthcare system. We were focusing, of course, on lifestyle and behavior, so it wasn't practicing medicine. But interested in this point at your comments. Of course, you've been involved. You were chief science officer at a major healthcare system for a number of years. And how are you thinking now about getting this sort of project, which if it is the future of medicine, how does that jive with the current healthcare system that we have, and how are we going to persuade those stakeholders, or do we really need to pursue it in a parallel path outside of the existing health system?
No, I think we have to actually attack it within the context of the current healthcare system. I think the really important idea in catalyzing fundamental paradigm changes and things like healthcare is to, within the system, find some CEO leaders who are themselves willing to think about pioneering the future of healthcare. And Phenome Health is exploring with a healthcare system now exactly this kind of role. The CEO is, I think, a real visionary. He is excited about the idea that he could really catalyze a fundamental change in healthcare. The healthcare system he's a part of integrates together the payer function with the provider function. And the initial cost savings from data-driven health are going to go to the payer side of the equation. So if they're integrated together, both benefit with the cost savings that comes. And of course, it always helps if you have money to catalyze change. And I think this system does have financial opportunities for catalyzing the change. But my argument is, after a 5-year period or so, we'll begin to see such a dramatic change in the health of their patients and in the enthusiasm of their physicians for a new kind of medicine, that they'll be a leader and others will be forced. in defense to realize that there is a much better way of generating health for individuals than through a sickness system, the sickness system we have today.
Yeah, no, I think that grassroots building up, and of course we've seen that some with the functional medicine and lifestyle and systems medicine approaches that individual practitioners are bringing to the fore, which is sort of forcing a change from the bottom up as well. Last question, I'm going to circle back to where we started, which is the idea of mentorship and how we really support each other and we build the scientists of the future based on the ideas and the characteristics that we can model. So what advice do you have for any students or postdocs or junior faculty who may be listening to the webinar today that are wanting to pursue a career in systems biology or systems medicine, so that they can avoid the trap of reductionism, which as we talked about in the beginning, even in systems biology and certainly in the silos of systems medicine, we're seeing today. So how do we encourage this new generation of scientists that are going to be our future in systems biology and systems medicine to really pursue the path of systems thinking?
Well, I would say there are a number of things that are important. First, for any student of biology, it is mandatory that you learn in a very deep way about some aspect of biology, that you get into the complexity, that you understand the nitty-gritty of the field. In molecular immunology, when I was growing up as a young assistant professor, that was a marvelous example because we were deciphering at that time mechanisms of antibody diversity, and that required reconstructing what happened in T cells and B cells. So, but understanding very deeply one aspect of biology is really important. I think a second thing that's really important is you really have to give yourself the tools, and these are understanding the technologies that let you make the measurements of appropriate types, whether it's DNA, RNA, or proteins, whether it's imaging kinds of things, is absolutely functional. And of course, understanding AI and the tools it gives you. I mean, I think everybody is going to be using large language models in fundamental ways to do their science in the future, and getting started now and early is, I think, a really important aspect of that. And being able to apply on a bigger scale your problems in biology through knowledge graphs, through digital twins, through large language models. I think that is going to become increasingly important in biology. But I think the heart of dealing with reductionism is really understanding what the essence of systems biology is. And it's that systems biology operates at a whole series of hierarchical levels, at the molecular level, at the level of cells, at the level of organs within an organism, and at the level of the organism itself. It operates at all of those levels. And basically what the integration of different data types lets you do is understand the relationship of these different levels as you bring together the data through appropriate kinds of integration. And I think if people do that, they won't take the much narrower view of systems biology, which is perfectly legitimate to focus on some very small aspect of systems biology. So I would argue reductionism is not at all a part of systems biology if you understand the totality of what systems biology means and the requirements it lays down on scientists and so forth. And that's what we have to have students prepared for. And then they can move into and use any kinds of tools, that is measurement, imaging, AI, whatever they are, in the context of this very big thinking, this holistic and global thinking about solving problems.
Yeah, absolutely. And I would even add, and this is certainly going to be a focus of our systems concepts journal, in addition to those levels within the organism, you also have the levels of the environment and the behavior which influence the organisms, and then the regulatory and the policy factors which can influence the behavior in the environment and ultimately the organisms all the way down to the molecular level. So it is really grasping that whole picture.
Agree totally with that.
Yeah.
Yeah,
Okay.
absolutely.
Well, Lee, thank you so much. I think this has been a very thought-provoking discussion. Lots here to digest. And I know we have some questions from the audience, so I am going to turn it back over to Sean right now to throw some of those questions your way.
Okay.
Thank you.
Excellent. Thank you. And that was a wonderful discussion and great insight to the history of systems biology and some of the Human Genome Project that led to a lot of the advances that we're seeing today, and maybe don't all have the right understanding of how we got here. And so that was a great education. I appreciate it. A few questions that have come your way. Number one, do you have any advice on how junior mid-level employees can promote systems thinking and innovation within their organization?
Well, I think the best way to do that is to set up seminars that begin to explore with people who are interested the basic precepts of things. I think beginning to educate people around you as to what systems thinking is, and more important, what it can do and the kinds of problems it can solve, I think is a way to go about it. There is a deep reductionism that is buried in biology still today. There is skepticism, even now, I think, about some aspects of systems biology, but the spectacular aspects of the problems it solved are now evident, and bringing them to the attention of your colleagues, I think, is a very powerful way to go about doing it. Of course, if you're in a position to run a lab yourself and start choosing problems that will be susceptible to the tools that you have available from a systems point of view, that's a more explicit way of doing it. But I think the education is a more global way. I remember when I was an undergraduate at Caltech, a graduate student at Caltech in biology, there were 8 of us students, and we set up a journal club that ended up being attended by faculty members and all sorts of things, where we explored all sorts of new dimensions of biology and so forth. And I think those approaches can be very powerful.
Excellent. Thank you. Next question takes a different approach, and there's a couple of follow-ups I'm going to summarize in one question. What are the possible applications of fractal geometry in cancer research in the framework of systems biology? And the individual also asks about fractional calculus and the concept of self-organization, again, within the context of cancer research in the framework of systems biology.
Well, I have to say, first off, I don't necessarily know very much about fractal mathematics and so forth. But what I will say is, for all the mathematical tools we've looked at in the context of biology, virtually all of them can be fashioned to do classification and analysis in biology. And I'm sure that these fractal approaches to things will be able to do exactly the same kind of thing. The key thing with new types of mathematics is figuring out how you can transform digital and imaging data into a language that new technology can digest and assess. Okay. And a very nice example of this is the large language models that we're dealing with now. They're very good at language. They aren't good at digital information from proteins and genes and all these things. So we've had to learn, and we're in the process of learning, how to convert the digital information through knowledge graphs into language information that then can be analyzed. And so you may have to manipulate things, but I think virtually all fields of mathematics could, in principle, lend to the analysis of biology's complexity is so infinite, you'll always have problems you can attack.
Indeed, thank you. The next question moves us back to the academy. And do you have any thoughts on how we can move academic culture towards a more inclusive default rather than the differential judgment of scientists performing method development and those doing hypothesis-driven science?
Well, I mean, that's a complicated question, and I don't know whether we're talking about the racial diversity and inequalities we're discussing there, or whether we're talking about the technologies. But as to the former, I think a really key aspect of biology in the future is that the data be gathered from individuals that represent the populations they serve. What is utterly clear from genome analyses is there are strikingly different responses to disease in individuals of different racial origins. And it's absolutely essential we know the genomes and the correlations of these differences so we can deal effectively with people. And the big fault, I think, is in the lack of diversity is it's a lack of data diversity. And that is something we can solve. And we actually, in this million-person project I've proposed as a genome-phenome project, a second genome project, I think we plan to have a diversity that reflects the racial diversity in the US so we can deal with those kinds of problems. Now, with regard to provincialism in tools and strategies for systems biology, again, I think you get back to needing to do educational programs and creating really effective and powerful courses in systems biology, I think is absolutely essential for all first-class institutions. And these days, most of them that I know of have really done that. But being able to reach out and talk about the tools, I think, and whether they're measurement tools or imaging tools or AI tools is really critical. And having the students in a general sense really understand those things and their relevance to problems they may become interested in is absolutely critical.
Wonderful, very insightful. Jumping over to the industry and life sciences side, the next question is, why isn't Big Pharma adopting systems biology with an open mind? And when do you think the computational systems biology will be used to deliver a success story in the drug discovery and development pipeline?
Well, I think Big Pharma is unbelievably conservative. And they have formulas for generating drugs, and generating drugs are the currency of the kingdom. And so that's what Big Pharma is all about. And I think they're reluctant to fiddle on the fringes of complexity. And one reason is Big Pharma is just now beginning to get away from the idea they'd like a pill for each disease. And that means everybody that has diabetes needs a pill, or everybody with Alzheimer's needs a pill. And they're just coming to an understanding that all of these major chronic diseases have subtypes, and the subtypes are very different from one another, and you're going to have to treat them in very different kinds of ways. And the key to getting at these subtypes are these systems approaches that are global and universal, and use big data then to be able to classify the subtype complexity of something like diabetes, which probably has 4 or 5 major different subtypes. I think that's one problem that pharma has. They're very, very slow to adopt tools that are even obviously very powerful. And look, there are occasional exceptions to this, but for the most part, pharma is very conservative. But I'll tell you another area that it's very conservative in is how it goes about making drugs that, if they're proteins these days, can cost $100,000 a year for a trial. And the most common types are, of course, cancer, focused on mutations and things like that. And this is supposedly the wonderful victory of precision medicine. But you know what these anti-cancer drugs do with most cancer patients is they work for 3 or 4 months, up to 9 months, even a year, but then the patient is back where they were before, only they've lost one additional option. So I read a New York Times article a couple of months ago that talked about a woman that had breast cancer that had gone through 8 such treatments. And you're miserable during these treatments. And it's not exactly, to me, a real victory for chronic disease like cancer. And I think where pharma really underestimates how the world is going to change is we're now developing through systems medicine and this data-driven medicine, the ability to detect the transition to chronic diseases years before they're clinically manifest. And it means we can think about therapy at an early stage when the disease complexity is very limited. And that's going to be the wave of the future. Early detection and prevention. It's 2 of the 4 P's and everything. So I think pharma, is enormously rich. It has a lot of money. It can afford to do things that often academics can't at all. But it is plugged into a very narrow view of how to do it. And I think that's going to have to change.
Wonderful. Very well said. Thank you for that insight and all the insights you have given us today. We're coming up on the hour. And so I am going to hand it off to Dr. Vodovotz now to give us a brief closing comment before I give you a preview in just a few seconds of what's going to come next. But again, Dr. Hood, thank you for being with us today. This has been a wonderful dialogue to kick off this new section of the journal. With that, I'll hand it off to Yoram.
Well, thanks, Sean. Yeah, as I'm so struck by really the great description and recounting and thought process that Dr. Hood has just expressed. I want to really, and what really struck me is how synergistic that is with what we're trying to do here in the journal. He said you sort of needed to start something new in order to be able to get this kind of new things done. That's sort of what we've tried to do with the journal. We've tried to maintain optimism and perseverance. We've set up these sections, especially this systems concepts section, which I think is going to have a lot to help with. Also, one of the things that Dr. Hood said, which is don't think as much about the methodology that you're using, but about the thought process that leads you to use one or another method. And that's really what we're trying to do with this journal, to use this as a platform to help, I think, recapture a thought process that maybe has gone a little bit by the wayside over the last few decades. And I really applaud you, Dr. Hood, for keeping the flame burning for this generalist and systems-oriented thought process. So again, thank you very much on behalf of the journal, and I'll turn it back to Jennifer and Sean.
Excellent. Again, well said, and thank you, Yoram. Thank you, Dr. Hood, and thank you to all of our guests who have been with us today and are still hanging in there a couple minutes over. I just want to close by giving you a highlight of something that we've just announced, which is the first special topic for this new section of the journal, and that is building systems thinking for machine intelligence in biology and medicine, inspired by by Dr. Hood's building systems thinking. I think there's a lot of new tools like LLMs and others that have been mentioned that are very good for what we can do in systems biology and systems medicine, but sometimes they're coming at us in one-offs and not being thought of in their own systems way. If we have multiple LLMs competing that give you different answers to the same question, or that you have one that gives you different answers to the same question across time, there may be difficulties in integrating these in really reliable ways in biology and medicine. So we're looking for concepts, and we're looking for policies, and we're looking for even real world efforts towards integrating and systems thinking across these different modalities of machine intelligence that we are applying to systems biology and systems medicine. And that's just up on the website today. Submission deadline for abstracts is the 29th of December. Manuscript submission deadline is 31 May of 2024. In addition to Jennifer and myself, Dr. Amy Elliott, who's one of our associate editors, will be the specialty topic editor for this call for papers. And I want to give a heads up and a preview to my own contribution on this end. I'm going to be putting forth a grand challenge. I call it Scientia Machina, using machine intelligence and computational trust for improved medical knowledge machine, looking at different layers of trust within the system of science and not trying to find one technology solution, but integrate different technology solutions into the human and organizational systems processes that we've refined over hundreds of years of science. I won't go into that to detail here, but we are going to have our next webinar in the coming months that will be announced, and this will be one of the topics that we cover. With that, I would like to thank you all and wish you a wonderful day. And please do consider if some of the work you're doing can be submitted to the journal. Thank you very much.